Maybe you know someone who's had a premature baby, and then you would know that the life of that child hangs in the balance. Maybe you've eaten some bad food at some point in time and you got sick, and perhaps you realise, you know what, that could have killed me. And maybe your sporting idol was done for the use of performance-enhancing drugs, and how did you feel? Well, today you're going to learn a little bit about separation science and understand that separation science supports every aspect of our modern life. In particular, I'm talking to you about pulse direct injection, which is a technique that we've developed at Western Sydney University that improves the efficiency, the speed, and the cost effectiveness of the separations industry. Now, separation science is perhaps the quintessential enabling science. It supports so many facets of our modern industry, from the pharmaceutical businesses to even workplace health and safety. Now, I'm not going to give you a lecture on what separation science is. You don't need that. But you do need to have a little bit of an understanding of how it fits into society. Everything we have in our real world is made up of complex mixtures of molecules. Separation science is about breaking that complexity into smaller entities. For example, the cup of coffee that you had this morning consists of hundreds of different compounds to give it flavour and aroma. Caffeine is just one of those compounds. Transportation of a sample through a separation device, as is shown on the screen, enables you to take the caffeine from that complexity. And that's what separation science is all about. It's not surprising then that the pharmaceutical industries rely so heavily on separation science. Imagine waking up in the morning with that migraine headache coming on, or you have got chronic pain. How do you deal with that? We think of nothing about opening up the cupboard, dragging out some pain relief, and away goes the headache. But what if we didn't have that? The pharmaceutical industry generates so many drugs that we take for granted. And with that, we have a raft of testing that needs to follow to make sure that what we take is safe and healthy for us. All the clinical trials we do involve the collection of samples to test whether or not the drugs that are built are actually serving the purpose for what they're intended. What if you can't take that Panadol, that aspirin, that Nurofen to remove the pain? Charlie Brown says it all. And that's what life is like if we don't have even basic pain relief. The little baby who's born prematurely, life hanging in the balance. Doctor administers a drug. What's the next dose? We need to test the response for the first dose. How is the baby tolerating that drug? Is it doing its job? Samples are collected from the baby, sent to the lab. They have to be processed and back to the doctor so that the doctor can administer the next dose. We need separations, and we need them to be done fast to get that information. The life literally hangs in the balance. The food industry is a massive user of separations to test the quality of our food, to make sure that what we consume is safe, and also to test the value of the food. We all know what happens when chemical toxins can enter into the environment of food, which is so easily done. We all can remember the melamine incident of baby formula. What are you feeding your kids? Separations are very important for testing food quality and maintaining health safety environments. Our sporting idols. Separations industry is very, very substantial in the area of sports. And every athlete is tested regularly, but they have to be done fast. Results need to come back to remove those who are using sports performance-enhancing drugs and so forth. Lance Armstrong, for example, is a classic example of evasion of that process. We also have to consider the athlete's safety as well and monitor regularly aspects that relate to sport. So they're just a few examples. So the current status of the separations industry is such that it spends billions of dollars annually worldwide supporting all the areas that require separations, from pharmaceuticals and healthcare to food, sports, and so forth. Now, the current processes undertaken with separations, they utilise what's called ultra-high performance liquid chromatography, coupled with mass spectral detection. That's classic current state of the art. It processes simple samples in time frames of minutes. And it does so at a cost estimate in a commercial lab of maybe $50 to $100 per sample, depending on how many you've got to test. But the future should be using our technology, pulse direct injection, with the same mass spec, but now we can process simple samples in seconds, not in minutes. Because we're more efficient, we're faster, and we're more cost effective, we can start bringing the cost of those analyses down. So we can start increasing the amount of analyses that are undertaken and do so cheaper. It changes the workflow practice, not just simplifying a single analysis. This is a workflow aspect as well. So why do we need pulse direct injection? Imagine if you didn't have that pain relief. Charlie Brown, I can't stand it. That's what life's like without the basic pharmaceuticals that we take for granted. A big area for pulse direct injection is in the process of optimizing the production of drugs to bring those costs down. What drugs don't we have because they're too expensive to manufacture? We don't know the answers to that. The little baby in the neonatal ward, relying on those test results to get back quickly. We need to be able to improve that process of an analysis to improve healthcare systems. And of course, melamine was picked up because the product crossed boundaries or borders. What's not being tested that we are in fact consuming toxic substances on a daily basis? Every athlete tested every event. That's what we need too. I lead a small scientific team at Western Sydney University. We're working with the Innovi's commercialisation team to bring pulse direct injection to the marketplace. Western Sydney University provides that industrial engagement connection between our science labs and Innovi's. So where are we at present? Well, we're currently working with 2 large pharma companies in California, and they want our prototypes into their system specifically for optimization of drug production processes. We've got a raft of publications that show the worth, and we have instrumentation manufacturers from the car industry in Adelaide wanting to build systems. We need partnerships to expand. We need beta labs to verify independently our claims. And of course, we're going to build lots of these systems. We need a distribution process in place. And we're after advisors who can think outside of the box that we're currently thinking inside.